Right. So Warren charged me with talking a little bit about reference panel construction for doing imputation, sort of on a practical level, both for research purposes. And I think with all the low pass stuff that Jesse talked about earlier and Matt, this is going to become an increasingly big part of what we think about is breed associations who want to do low pass sequencing and imputation at a high level. How are we choosing which animals go into these reference panels? What do these reference panels look like, and how do we maximize the utility of this extra information from imputation? And so a lot of this is going to be stuff. I need to say a thanks first to Bob Schnabel at the University of Missouri. Bob and I are messaging and calling back and forth about weekly, talking about this reference panel construction and doing imputation and stuff that we're continuing to work on even after I wrapped up my PhD at Mizzou. And I'll talk a fair bit about a study that we did, Bob, Jared, Jesse, and I, during my PhD, looking at chip level imputation and considerations that I think we can port over to how we do sequencing and building these imputation reference panels at the sequence level. And so in the beef industry and really across agrigenomics, right, we're very good at generating data that flows in to these genomic predictions, and it's revolutionized things, right? But I think that we're, a lot of people, including Ignacio Mistal, this is a paper from back in 2015 or something like that. Is genomic selection a mature technology, right? Are we plateauing on these traits that we have enough phenotypes for? Are we plateauing in our accuracies in genomic prediction? What are the next steps here, right? How do we move into that next generation of moving past just these mixed model equations, using our SNP chips to make this G matrix or the H matrix if you're doing single step, in that 50,000 marker set that we came up with back in the late 2000s has served us really well when it comes to doing genomic prediction, right? And there's been plenty of stuff. This is from a paper that Megan published back in 2012 that even subsetting that 50K marker set down even further, we can still capture that information to recapitulate those relationships between individuals genomically with a pretty limited set of markers. But when it comes to doing work in the space of identifying the causal genetic factors that play into these complex traits, when it comes to adding functional genetics into our predictions, which I think is a really rapidly evolving space, we need a much higher resolution than what these original SNP assays offered us. And I think that with imputation, again, whether that's from low pass or whether that's adding value to these really big existing chip databases, imputation allows us to fairly cheaply, if we've got a big bunch of sequenced animals, turn those 50,000 markers into a significant amount of additional information. And from that, if we can collect phenotypes and attach these imputed very high resolution genotypes, we can really start to dig into the functional biology that's underlying these complex traits. And there's been a barrage of research and work that shows that if we can accurately impute, some of the early studies that start looking at putting sequence imputed variants into genetic prediction algorithms, they weren't seeing a lot of increase in prediction accuracy, not a lot of extra utility by adding those markers. But I think that as we've gotten better at imputing, we've reduced some of that noise that exists due to imputation errors. We're starting to see in a lot of cases adding this really fine scale functional information into genomic predictions can help us, particularly on traits with complex genetic architectures. So as we move forward and think about this from a breed association genetic evaluation perspective, which is what we're talking about here, I think that in the future, the better that we can do at imputation, the more informative phenotypes we can collect and connect those two things together, we can really leverage that to make better genomic predictions at the end of the day for our producers and stakeholders. And the other thing that I think we're in an exciting time here is that the opportunity to do sequencing and leverage sequencing for genotyping is continuing to this cost is declining over time. And just this week, a new company, Ultima Genetics, comes out and they claim that they can do $100 full coverage genome, which is really an extraordinary development when we think about this. Where we've come from the announcement of the Human Genome Project, this $100 million plus project back in the 90s, to we're routinely sequencing animals for $300 to $400 now. And the ability to do that, I think, gives us a bunch of opportunities, both in the low pass realm, so using this to generate genotypes, but also on the imputation front, right? And not just for low pass, but also adding some value to do biological discovery in our big existing chip-based genomic data sets, because this sequencing is all of a sudden pretty affordable to do at a much larger scale than it was even back when I started grad school. That cost has continued to drop on down. And so as we think about these things, these are sort of the big reasons that I think that having these sequence imputed genotypes, doing these well, can have a really big effect on genetic evaluation. And a lot of this is my interest as a biologist, is trying to tease apart some of this biology and port that over into a genetic prediction, do that better, informing these things with some causal biology. All the work that's been done in functionally annotating these animal genomes. We have all of this information out there in lots of research that shows if we're able to account for this correctly, we're able to add some extra information to these genetic evaluations. And so again, there's lots of different ways we can slice and dice and think about putting these into a genetic evaluation. But integration past this, we have a high quality imputed data set of 100,000 individuals. Integrating that and choosing how we want to do that is going to be a constantly evolving area in this field, and something that is academics, we need to be good partners to breed associations, to service providers like Neogen and Genco. Understanding this biology and turning that over to y'all to put to work, I think is our big challenge here. And so I want to talk really briefly about imputation, what it is, and more specifically, the important part about imputation is developing these reference panels to adequately represent the population that you're trying to impute. And so the gist of imputation, which I'm sure most people in this room are familiar with, is that when we genotype on a SNP chip, we have a handful of markers that we've evenly spaced throughout the genome. We keep these at moderate to high frequency, so that we're able to pick up the maximum amount of variation on that limited subset of genotypes, right? But we also know that there's a lot of missingness in this, right? So if we've got cattle with 30 to 60 million SNPs, depending on where you're doing your SNP discovery, we're only representing a tiny fraction of the total diversity that exists across these animals. So the idea is that if we're trying to do some sort of an association test, and we are only genotyping 50,000 markers across the genome, we're getting a limited snapshot. We're likely to miss some of the stronger associations that might be there, but that just aren't genotyped on our assay or whatnot. And so the idea is that if we have a big set of reference haplotypes, so these are animals at a higher density than this original chip genotype, we can match up the haplotypes from this chip genotype, overlay it onto these haplotypes, and fill in the missing stuff that doesn't get typed on these chips or typed immediately by a low pass sequence. Whatever that imputation looks like, we're really just trying to match the patterns that exist in it and fill in the intervening stuff, right? And then what we're left with, of course, is this imputed data set. So these are statistically inferred genotypes based on a reference set of haplotypes. And the ultimate goal is hopefully that we're able to capture some of these signals that weren't immediately genotyped on that initial assay. And so the focus, again, like I said today, is going to be on this reference set of haplotypes. And the big takeaways that I want to really lay down is that we can impute things that we don't observe, right? We're not going to magically impute missing genotypes that aren't represented in this reference set of haplotypes. And so as we think about doing high quality imputation in our data sets, whatever that looks like, we have to do our best job of representing all the diversity that exists there in order to make sure that this imputation is high quality, because again, low quality imputation isn't particularly useful. That's just throwing noise into the mix there. And one thing that I wanted to just really drive home this idea of visualizing imputation. So I came up with this while I was sitting waiting for the bus in the El Paso airport. So this is a 47-mile drive, or a 47-minute drive from the El Paso airport to the convention center here. And so if we imagine that like a strand of DNA, right? This is 49.2 miles. Took us 47 minutes to get there. And we think about, I guess, looking out the window during this drive from El Paso to the convention center, okay? Using an unimputed chip genotype, so this is, I just did some back of the napkin math, so this might not all check out, but no peer review in this talk. But having an unimputed chip genotype is like getting a 1-second look out the window every 5 minutes that you're driving from El Paso up here to Las Cruces. The sequence level genotypes that we get through imputation, though, is like taking a 1-second look every 5 seconds. So the ability to understand the surroundings, see as much information as possible, right? If this is a genome, we're getting a much better picture of what's going on there because we're able to observe those little glances in the genome, imputing those SNPs at a much more frequent pace, right? So I don't know if that was a great visualization, but again, the resolution that we get through imputation, especially when we do it well, is really helpful when we start to try and dig down into some of these association signals. And this is from a paper that we published last year, and I really took the same region of the genome. This is about 3 megabases on the end of chromosome 23 in a GWAS. And it's the same data set, the same trait. The only thing that I've changed is the SNP set that gets used for each of these. So the top up here is the SNP 50. So sort of the standard thing. This is, I believe, in American Red Angus is where this population is. And this is the association on the markers that are on the SNP 50. This is from a data set of about 850K imputed markers, so 850,000. And this is what sequence imputed genotypes look like across here. And the really amazing thing, right, is that if we're trying to do any sort of association mapping with raw 50K data, it's really hard to, unless you're just right on the money, you've tagged something that's potentially causal through the discovery phase of making that assay, you're probably going to miss most of the association signatures as you look across the genome. And again, a much more higher resolution look in the 850,000 marker set, we're able to see this signature is actually kind of complex. So we have a really strong main peak here and a couple of sub-associations that we can't be super sure if these are actual associations or just little spurious things hovering above our significance threshold. But again, when we add this next level of information through accurate sequence imputed genotypes, you see that these are actually, we see some little smaller LD peaks over to the side of this main association. Again, allowing us to dig into these complex signatures with a much higher resolution because we're able to do imputation well. And again, if our goal is to do sort of SNP discovery, identifying informative markers, causal associations, doing imputation better is our best way of getting there. If we have messy imputation, we lose statistical power to detect those true associations. So this is from a human study where they're comparing the power to detect these the various effect size SNPs in this association or in this association study at both common and rare minor allele frequencies. And so what you'll see is that as they sort of increase the quality of their imputation reference panel, here is what they're comparing, and comparing to this gold standard of whole genome sequence where we assume that everything that we're genotyping is perfect, right? Our goal here, right, is to reduce this area between these curves, right? We want our imputation to be as close to what we would get as if we'd done a 15x sequencing run on all of our samples, right? And so again, all of this imputation quality and power, of course, is minor allele frequency dependent. So we get into these rare alleles of very small effect, and it's extremely hard for us to detect these, particularly if there's a bunch of noise that's been introduced through poor imputation. So again, our goal of building these reference panels and doing it in a constructive and organized manner is to make sure that we're representing as much diversity here so that we can kind of close that gap between pretty good imputation, really good imputation in getting up there towards sort of the gold standard of high accuracy, high coverage genotypes on everything. And so an accurate imputation needs a really large set of accurate reference haplotypes, right? If you don't observe it, you're not going to be able to impute it. And Bob Schnabel always says that it's crap in, crap out. If you don't do a good job of calling your genotypes, right, you're not going to, if this reference panel isn't both representing the diversity, or we've done a poor job of actually calling the genotypes that exist in there, we're not going to do a good job of imputing at the end of the day. And again, having a good reference genome and understanding recombination is also important for doing imputation at a high quality. And one of the questions that I think we have to ask, if I'm a researcher who wants to build on these imputation resources, I'm a breed association who's thinking about low pass. We need to sequence some animals so that we're represented in this big imputation reference. Coverage consideration is always the thing, right? How high of coverage, how confident do I need to be in these sequenced animals that'll go into the reference panel in order to make that work? And this is a figure from Bob at Mizzou, and he did a bunch of work looking at trios, so sire, dam, offspring, and looking at the number of Mendelian errors that occur in these trios based on coverage. And what you'll see is that this really quick drop off happens right around the 10x genome coverage of these animals, right? So there's a fair number, probably 8 to 10 times as many Mendelian discordances up here in the 5x sequenced animals compared with these animals that have 10x plus coverage. So as we think about developing these resources from a coverage standpoint, there's a really clear indication that doing 10x plus is going to benefit us just in the quality of calls that we're able to get in these reference individuals that we're sequencing. And then also, I think it's important, and I'll talk a little bit about imputation accuracy here, but measuring accuracy there's a lot of different ways we can do it, and they're going to give you different answers depending on which sort of a metric you're using. And the big one that I think a lot about is that concordance is helpful, but it's not a perfect measure of how well you're imputing a genotype, right? This is just saying there's a SNP here. Did I impute it correctly? Yes or no? And the issue that arises here is that when we're at really, really low minor allele frequencies, so if we've got a variant that's 0.5% frequent, we're going to impute that, 99.5% of the time correctly, just because its allele frequency is really low. So doing something like a correlation, or what I'll present here are imputation quality statistics. Take that sort of, I guess, random chance that you're going to get something right due to it being very rare, out of the equation, right? And it does a better job of evaluating that actual imputation accuracy than just that simple yes or no, did I get it right? And then the other thing too is that there's a lot of value in doing truth set validation. So having animals that have, either a high density genotype, a high density true genotype, or a sequenced individual at high coverage that we can downsample and ask how well are we actually imputing these genotypes, versus just relying on internal values that software spit out. I've done some work looking at these, and there can be a fair bit of discrepancy from what a software is able to estimate versus what happens when we look at actual true data. And I will put a plug in here that Bob is looking for additional trios to sequence. He's got some extra money to sequence trios. So if you're a breed association person and you have some trios of interest that you'd be interested in having sequence data on, give me a shout, and we can get together. I think a good opportunity to give us more animals to test imputation accuracy and quality, as well as some of the base calling work that Bob is doing at Mizzou, while also adding to your genomic resources through that sort of avenue. So just a little aside there. And then the big question, and the thing that I think your big take home here today, is that we can build reference panels to do imputation in a couple main ways, right? We can make a breed specific reference panel. We could even make a beef specific reference panel, or a dairy specific reference panel, or and this is different from what Ben talked about, but along the same lines, right? Represent all of the haplotypic diversity across all of the cattle that we have access to, and use that one reference panel as an imputation reference for all the animals that come through, right? And so we did a little bit of work on this during my PhD, and I'm going to present these results. My disclaimer, I guess, is that they're all high density chip imputation, and we're extrapolating this out that when we go and move into sequence, and I've got a grad student primed and ready to start analyzing some of this. When we move to sequence, we'll see similar trends when it comes to imputation quality based on the breed specific or multi-breed references. And the reason why I think that this multi-breed reference works well is that it's hard for you to impute something that you don't observe, right? I've said this about 4 times, but that's the big thing, right? And we know that especially in open herd book breeds, there's a lot of outside of the labeled breed of haplotypes that are floating around in there, right? And our only opportunity to impute those is if we're able to observe those. And then again, a breed label doesn't guarantee shared or not shared haplotypes, right? So we've got, I'm thinking back to the database at Mizzou and similar to stuff that we've looked at in genotypes in my lab at Tennessee, is that the breed label that gets slapped on an animal doesn't necessarily mean that because he's called Angus, he might be black-hided, but there might be a lot of different stuff going on there. So keeping in mind that we don't want to pigeonhole ourselves with a limited imputation reference just because of an arbitrary label that's gotten put on a sample of some sort. Just to share a little bit of data here in the last little bit that I have to talk, diverse samples need a diverse imputation reference panel. And so this is looking at a group of about 50 Gelbvieh animals that we had high density chip genotypes on, and we downsample them to a 50K set of SNPs. We do imputation and we ask how high a quality is the imputation as we look across the minor allele frequency spectrum, right? So these are very common alleles out here, and these are more rare alleles as we move back over to the left of this graph. And what you see is that in green is a Gelbvieh specific reference. We were only using animals in that reference that were labeled as Gelbvieh in the database, whereas this multi-breed reference uses all of the animals that we have access to. And what you see is that in the common alleles, we do a pretty good job of imputing with both of these references. But the real kicker comes here in the rarer side of the minor allele frequency spectrum, where we see a significantly higher advantage to this multi-breed reference as compared to the within breed reference when we're imputing these rare alleles. And again, this is just because these things are probably segregating. If they're rare at a low frequency in the larger cattle population, you're much more likely to observe this, this rare, maybe Angus haplotype in the multi-breed reference than in a restricted Gelbvieh breed-specific reference. Again, due to the nature of Gelbvieh being an open herd book breed. And the idea that imputation is just pattern matching and you can't impute what you don't see. We look at these Gelbvieh individuals and we ask how many imputation errors do we make on a per individual basis. And so this is the number of errors. Each dot is an individual animal, the number of errors there on the y-axis that we make in imputation. And what you'll see is that with using a breed-specific reference, there's a handful of these individuals that impute pretty poorly, okay? But when we add in the composite reference, we see that the number of errors decreases quite a bit for these individuals that we impute poorly. But the other thing to take away is that by adding in this composite reference, we don't do any worse on these animals that already got imputed well using the breed-specific reference. So this is one of these situations where you can't do any harm by throwing more haplotype diversity into an imputation reference. You can only do better. The animals that are going to impute well on the breed-specific reference are going to do just as well as you move over into a composite reference. And then this idea that representation matters. So this is looking across the test animals that we evaluated their imputation accuracy in. And you'll see that for breeds that are well represented in the imputation reference panel, we impute really well, which is no surprise. A few weird little stragglers in Angus that, again, when we dig in past that label of Angus as their breed, there's some weird crossbred animals that got in the data set, but labeled as Angus, right? But otherwise, you look at breeds like Gelbvieh, Hereford, Holstein, all of these well-represented breeds in the reference, we impute quite well. Whereas when we move into breeds like Gyr or Romagnola that are much more sparsely populated in our imputation reference, we don't do nearly as good a job of imputing them, and even worse on a breed like Nadama, an African breed where we're just not representing those in the high accurate or the higher density reference that we use. The other thing, and this is looking across the genome at places where we make imputation errors. And so this tells me that there's a lot of room for improvement. And since this paper was published, I know that Bob has gone back and dug into a number of these loci and fixed some misplaced probes in the reference. There are a number of little issues and some imputation software things that I think you could tweak and fix some of these up. Again, you'll see many of these places with higher numbers of errors are at the ends of chromosomes. Again, either errors in the assembly, which we're capable of fixing, misplaced probe sequences, so they're genotyping at the wrong spot, out of their correct genomic context. But again, lots of ways for us to continue improving the accuracy with which we do imputation. And so the other big question, I think, is as we go out, if I'm a breed association or if I'm an academic who's trying to make this imputation reference panel better, how do we choose animals that get to go in the reference? And I think that there's the easy answer is, the highest progeny number. We want to represent the haplotypes that make up our breed as well as we can. I would argue that in John Beaver, my contemporary at the University of Tennessee, would say that once you reach a certain threshold of calf sired, we should probably have a whole genome sequence on these bulls, just in case a genetic defect pops up, something along those lines. But again, I think that that's a big consideration. But again, also being conscientious of, do we need to sequence a sire-son combination? If we've already got the high use sire in the imputation reference, how much additional benefit do we get from adding his son in there, right? But then I also think that with the large genomic data sets, particularly chip genomic data sets that we're constantly generating now, we can utilize that genotype data to drive the sequencing that we do, right? See which haplotypes we're observing at high frequencies in the population, but that aren't represented by our already sequenced animals in the imputation reference. Utilize that genotype data to inform which animals get sequenced. So I think there's lots of ways to do this. We've done a little work with the American Simmental Association in picking out some high use sires that would add into an imputation reference down the line, and something that will continue to sort of fine tune how we do that animal collection. And so as far as future opportunities, I think that there's some really exciting stuff going on. Again, we're glad to have Ben here and talking about the pangenome, and I think that when it comes to doing pangenome aware imputation down the line, there's going to be a lot of value when we can take a resource like a pangenome that represents a ton of genetic diversity across Bos taurus and Bos indicus, and then layer on the power of our really large genomic data sets. We can add a lot of really interesting information there. And then software development in the genotype caller space, which again is much more Bob Schnabel's thing than mine, but I think there's some really interesting developments coming down the line that'll make us better able to do all of these things, whether it's calling genotypes or imputing. And I put rare alleles on there for a reason that I can't remember at the moment. So my recommendations, I guess, and I'll hop off my soapbox right after this, but we need to sequence more animals, right? I think that we understand that the low pass wave is coming in, right? And imputation is at the core of that. And we also have a ton of resources outside of low pass sequencing that would benefit from imputation from a research and discovery perspective. And so being intentional about trying to represent those novel haplotypes is going to be pretty crucial, and making that data public and accessible, some sort of a large resource that we're all able to draw on, not hiding this behind walls where we're not able to pull that extra information from one another. Again, this minimum coverage suggestion of 10x, I think that there's been enough data to show that that's a pretty good threshold for inclusion into these imputation references. And not only doing this imputation, but we need to go back and evaluate imputation accuracy and make sure that we're doing things right and figuring out the things that we can tweak to possibly improve this. And then also, I think that the really exciting thing isn't necessarily just imputation, but when we have an extra 30 million SNPs, what do we do with that extra information? What sort of biology can we start to understand? How can we turn that into actual transferable stuff in the form of a genetic evaluation, which I think is the 30 million SNP question there. So I'll leave you with my my ill-formatted takeaways here and contact information, and I'm happy to take questions here or over at the panel as we wrap things up.